Good morning. Today's topic of discussion is glycogen metabolism. Glycogen is an important storage form of energy in animals, just like starch as in storage form in the plants. Stored in the liver around 100 grams, that is equal to 6 to 8%. And in the muscles, the storage is 400 grams, that is equal to 1 to 2%. The total quantity of muscle glycogen is more than the liver glycogen because of the larger muscle mass. So all the enzymes related to the glycogen metabolisms are cytoplasmic. Literally, the glycogenesis is the synthesis of glycogen from glucose. The synthesis happens in the cytosol and it requires energy in the form of ATP and UTP. The steps of glycogenesis involve the first is activation of glucose and synthesis of UDP glucose. Second is the synthesis of primer to initiate the glycogen synthesis. Third is the elongation of glycogen chain by glycogen synthase. And fourth is the formation of branches in glycogen. So we can see in this structure in flow sheet diagram, this is the glucose. Glucose is converted up into glucose 6-phosphate. In the well-fed state, the glucokinase is the enzyme which converts the glucose into the glucose 6-phosphate, and glucose 6-phosphate via the mutase enzyme, phosphoglucomutase enzyme, it converts up into the glucose 1-phosphate. Further, the UDP is utilized and it converts the glucose into UDP glucose. One pyrophosphate molecule is lost here. So this UDP glucose is acting as a monomer, all set to start the synthesis. Then the glycogen is a protein which form the platform over which the synthesis of glycogen takes place. So via the hydroxyl group, there will be attachment of this glucose, and so glycogen initiator synthase enzyme, it leads to the attachment of glucose sequentially and leading to the formation of glycogen primer, which is having 3 to 5 monomeric units attached with the glycogen. Once the proper platform is being formed, further the UDP glucose keep on adding one by one, and there is a formation of long chain of glycogen, linear chain of the glycogen. And this, once it is 10 to 16 units long, then there is a enzyme glucosyl alpha 4-6 transferase, which picks up 4 to 6 unit of the glucose chain from the termini, and it attaches at the middle around 6 to 7th number of this glucose unit via the alpha 1-6 bond. So this is how the branching, this makes the branching point. And then further, the glycogen synthase keep on adding the glucose unit sequentially here and here also. Once again, it is 10 to 15 chains long. Again, there will be a picking of 4 to 6 units, and then they reach to the branching point and they make the branch, and another chain is now starts forming. So elongation by glycogen synthase forming the alpha 1-4 bonds. And then branching by glycosyl 4-6 transferase and alpha 1-6 bonds. And finally, these steps go and go on, and these lead to the glycogen molecule, which is highly branched structure. You can see it here. This is the branch structure of glycogen. So many linear chains, and they are branched and rebranched again. So this is the complex structure of glycogen, which is totally different from that of starch, which is maximally linear, and but both of them are consist of glucose. Now, glycogenolysis. Glycogenolysis is basically the breakdown of glycogen into the glucose monomeric unit. So you can see it here in the glycogen, two types of bonds are there. Alpha 1-4 linkages are there, and the alpha 1-6 linkages are there. There are three different enzymes are working against the breakdown of glycogen. Main enzyme, main glycogenolytic enzyme is glycogen phosphorylase. And then another enzyme is transferase, and then we have a debranching enzyme. All these work in coordination, leading to the complete conversion of glycogen into the glucose 1-phosphate entities. So see one by one. So this is the glycogen phosphorylase. This is the first enzyme the glycogen has to meet while its lysis has to happen. So it breaks down the alpha 1,4 linkage and leading to the release of glucose 1-phosphate units sequentially, and then breakdown keeps going on till the trisaccharide unit is left attached at the branching point. So till this point there is a after this point there is no breakdown, and then comes the action of another enzyme, that is the transferase enzyme. It picks up this particular trisaccharide unit and shift it to the long chain side. So attachment to this chain and lengthening of chain is there. Then the debranching enzyme will break down this glucose 1,6 glycosidic linkage, leading to the release of glucose unit. Then then again the glycogen phosphorylase starts acting and keeps chopping off the glucose units in the form of glucose 1-phosphate. Now, regulation of glycogen metabolism. There are two important enzymes, glycogen synthase, which is the glycogenic enzyme, and then second enzyme is the glycogen phosphorylase, which is the glycogenolytic enzyme. The two states are important in the regulation. One is the dephosphorylated and another one is the phosphorylated state. And the two important hormone, insulin and glucagon. Insulin always favors the glycogenesis while the glucagon favors the glycogenolysis. So we will see it here in this particular diagram. This is the glycogen and glucose with the help of enzyme glycogen phosphorylase, it converts up into the glucose 1-phosphate. Again, the glucose 1-phosphate with the help of enzyme glycogen synthase, it can revert, it can be converted into the glycogen. So this cycle goes on, and how it is being regulated. So there are various inhibitory factors. The inhibitors of glycogen phosphorylase is glucose 6-phosphate, ATP, and the presence of liver glucose, then muscle calcium, and the AMP has the stimulatory effect onto the glycogen phosphorylase. While the glycogen synthase is being stimulated by the glucose 6-phosphate. Means once the abundance of glucose or glucose phosphate 6-phosphate is there, it always prefers to be converted into the glycogen and stored. So we can see here what is the role of calcium in muscles. During the muscle contraction, the calcium is released from the sarcoplasmic reticulum, and calcium 2 plus binds to the calmodulin unit of phosphorylase kinase B, activating it without phosphorylation. And phosphorylase kinase can then be activate the glycogen phosphorylase, causing the glycogen degradation. So we can better understand here at this point. See, this is the glucagon bound to the glucagon receptor, and this is the membrane. This is the membrane bound receptor. So once there is an binding to the receptor, internal signal transduction happens within the cell. So GTP units get activated, which leads to the activation of adenyl cyclase, which converts ATP into the cyclic AMP, and this cyclic AMP converts the inactive form of protein kinases into the active form of protein kinase. And this active kinase, what it does, it converts the glycogen phosphorylase kinase B, which is in the inactive form, into the activated form, that is the glycogen phosphorylase kinase A form. And that means the glycogen is always active in the phosphorylated form, while it is inactivated in the dephosphorylated form. So now the glycogen is activated. Once it is activated, this phosphorylase enzyme starts the glycogenolysis. So what is happening here? It prefers the glycogenolysis, means breakdown of glycogen to glucose. Then in the presence of insulin, insulin is has a stimulatory effect onto the protein phosphatase enzyme, and it always converts or prefers to convert this active form of glycogen into the inactive form. So what will happen? There is a, this enzyme is now activated in the dephosphorylated state. So glycogen phosphorylase kinase B is now inactive because it is now dephosphorylated in the presence of insulin. So insulin always prefers the synthesis of glycogen, while the glucagon always inhibits the synthesis of glycogen, while it prefers the breakdown of glycogen. Its effort is always to increase the concentration of glucose in the blood, as it is an hyperglycemic, while insulin is a hypoglycemic, right? So, so this cycle goes and goes on. So, role of AMP in the muscles. In muscle, under extreme conditions of anoxia and depletion of ATP, AMP is activated and it activates the glycogen phosphorylase B without it being phosphorylated. So this is another way to increase the concentration of glucose by increasing the glycogenolysis process.